Hello, children. Coming to the sexual reproduction in flowering plants. In the previous video, we have learned up to the formation of zygote. And we learned that the zygote develops into embryo. There, if you remember again, the events of the sexual reproduction in flowering plants. First, we have learned, children, the first is pre-fertilization, then fertilization. In pre-fertilization, we have learnt about gametogenesis and gamete transfer. In fertilization, we have learnt about the double fertilization, that is, and triple fusion. These two we have completed, children. Now we should come to the third step, that is the post-fertilization events. So what happens after fertilization? What are the changes that will take place in the embryo sac? Let us see, children, here in the post-fertilization events. We should see, first we should know that the formation of endosperm. Actually, you know what is endosperm? What is the function of endosperm? If you think it is used, it provides nutrition for developing embryo. So, in plants, sexual reproduction, the development of endosperm precedes embryo development. There is a sentence in your textbook, children. If you say the sentence, the development of endosperm precedes embryo development. Why? The reason is endosperm provides nutrition for the developing embryo. That is why, for assured nutrition for the development of embryo, the embryo development से पहले क्या होता है? The endosperm will be formed. After formation of a little endosperm starts, then only the development of embryo will start. So, how the endosperm is formed now? If you remember the triple fusion, children, two polar nuclei in the embryo sac fuses with one male gamete to form PEN, primary endosperm nucleus. So that is the beginning for the formation of endosperm. And this primary endosperm nucleus is formed inside the central cell. So now the central cell is no more called as central cell. It is called as primary endosperm cell. So in the primary endosperm cell, the primary endosperm nucleus starts dividing by mitosis and forms a large number of free nuclei. Then the endosperm is called free nuclear endosperm. Later, the cell walls will be formed and it will develop a second stage as cellular endosperm. So the free nuclear endosperm, example you can take, to some cells will be, some nuclei will be free. That is, if you see the coconut, children, when you drink the coconut water, that coconut water itself is the free nuclear endosperm. So later on it will develop slowly. If you see, the entire water will be disappeared and only the kernel will be formed. So what happens at the time? The cell walls will be formed and will become a cellular endosperm. It is called as the white part, the solid part of the coconut, that is called as the kernel. So, children, you will be getting a question, why the development of endosperm precedes the development of embryo? Explain. Or why, reason. So this will be coming in the exam as a question. And cellular endosperm and free nuclear endosperm. These two you have to remember in coconut. Coming to the embryo. Now you have learnt about endosperm. Now let us see where the embryo is formed. Where is the fertilization of syngamy took place? The male gamete fuses with the egg near the micropylar region where the egg apparatus is there, and it will form into zygote. Then embryogenesis. So the embryo which is formed in the embryo sac, then what happens? Mature embryo sac. If you see, children, only the primary endosperm cell will be there, whereas the antipodals and synergids will be degenerating. That diagram is important. Mature embryo sac after fertilization. So here the embryogenesis, if you observe the diagram, children, the division will be continuing in the zygote and it will be developing into different forms of embryo. That is, the first one is the proembryo, then globular embryo, then heart-shaped embryo, then you can see the mature embryo. So these are all done with the division, that is mitosis. So coming to the structure of embryo. Here the embryo is generally having an embryonal axis. For assumption, we see an embryonal axis which also contains two cotyledons if it is a dicot and one cotyledon if it is a monocot embryo. The embryonal axis, why we are taking as a measure for learning above the cotyledons, the presence of the part of the embryonal axis above the cotyledons, we call it as epicotyl because it is above cotyledons, epicotyl, and this will be terminating or developing into plumule. You know, children, radicle and plumule you have learnt in 11th class also. The plumule is the future shoot or the stem. Below the cotyledons, the embryonal axis is called hypocotyl, which will be developing into future radicle. And this radicle is nothing but the root which is protected by root cap. So if you observe the monocot embryo, children, it is having only one cotyledon. That one cotyledon is called scutellum, which is present at the lower end of the embryonal axis. There you can see again radicle, which is protected by coleorhiza. The radicle is protected by coleorhiza. And above scutellum, you can see again the shoot apex, that is above the epicotyl. There is shoot apex protected by coleoptile, that is a stem tip. Future stem is protected by coleoptile and future root radicle is protected by coleorhiza. So this is the difference, children, between monocot embryo and dicot embryo. If you see the dicot embryo, there are two cotyledons. If you observe the monocot embryo, there is only one cotyledon, which is also called as scutellum. In the dicot embryo, above the embryonal axis above the cotyledons will develop into is known as epicotyl, which terminates into plumule, which is the future stem. Below the cotyledons, the embryonal axis is called hypocotyl, which develops into radicle, that is a future root, which is also covered by root cap. If it is the structure of monocot embryo, if you observe, the scutellum is called, the cotyledon is called as a scutellum, and the lower end of the embryonal axis is a radicle protected by coleorhiza. And above the scutellum is epicotyl, which is containing the shoot apex and the leaf primordia, and the shoot apex is protected by coleoptile. This is these two diagrams, the monocot embryo, dicot embryo, are very important, children, for exam. So you have to practice the diagrams. And coming to the seed, because we are learning now, children, about the post-fertilization events. Now we have seen the development of embryo, we have seen, and structure of embryo we have studied. Now seed is nothing but fertilized ovule. You know that ovary contains ovule contains embryo sac, which is a female gametophyte. Now the ovule, after fertilization, the ovule only develops into seed. So that is why we call the seed, we can also call the seed as fertilized ovule. So what are the parts of the seed? The seed is having two integuments, two layers of the outside. These are nothing but, this seed coat is nothing but the integuments of the ovule. If you remember back again, if you recollect, children, ovule is having two integuments which contain the special tissue known as nucellus. So these integuments of the ovule will develop into seed coat. Then the seed coat will protect inside the seed coat, two layers if you take out, there are cotyledons, and inside the cotyledons there is an embryo. And the seed coat, where the seed coat is there, inside the seed coat again there is a small pore. That is nothing but the micropyle of the ovule. The small micropyle of the ovule remains as a small pore in the seed coat, which is helping in the entry of oxygen and water. Because before seed germination, it needs to absorb oxygen and also water, and it will help in germination or development of the germination of the seed. So, entry of oxygen and water are helped by the small pore in the seed coat. The small pore is nothing but micropyle of the ovule. Now, the stored food, how the embryo inside the seed is developing because of the stored food inside the seed. So, this stored food, whether the embryo development, that endosperm, we are calling it as endosperm. And if the endosperm is still persisting or it is exhausted during the development of embryo. So based on this, the presence or absence or finished, then it is called, the seeds are classified into two types. One is albuminous seed and other are non-albuminous seed. In albuminous seed, children, they contain a part of endosperm, a residual endosperm. A small amount of endosperm is still remaining in the seeds. Actually, it will be completed there only within the embryo development only, it will be used up, but still a small amount is remaining in the seed, then we call it as albuminous seed. So, here the other examples, children, wheat and maize. Non-albuminous, there is no residual endosperm. The entire endosperm is consumed during the development of embryo. Then that is called non-albuminous. Example, peas, groundnut and all. These are very important for 2-mark question, children. Seeds and what about the albuminous and non-albuminous seeds? Albuminous contain the residual endosperm, and whereas non-albuminous, there is no residual or leftover endosperm. Entire endosperm is consumed. And another is also there, children, like perisperm, very important for 1 mark. It also provides nourishment in the seed, but perisperm is not connected with endosperm, but it is the remnants of nucellus. Children, nucellus already I made you to recollect again, which is there in the integuments of the ovule. This nucellus is still remaining as a residue in the seed. We call it as perisperm. So, perisperm is nothing but residual nucellus. Next. In coming to the seeds we know, children, we will keep it for a long time and dry under dry conditions, because moisture will help in the development of microbes which spoil the seeds. So generally the seeds will have very less moisture and slows down the metabolic activity. Whenever you buy the seeds and all, you keep rajma or kabul chana and other things we bring and keep, and they are very dry. Can you eat them like that? First, before eating, we have to soak them in water, so that they absorb water and they become swollen up and then they become soft. And you take out and leave it in the soil, then you can see in the moist conditions it will start germinating. So until it gets the favorable conditions, the metabolic activities of the embryo in the seed remain suspended or slows down. This condition we call it as dormancy. dormancy, dormancy. So germination of seed takes place only in favorable conditions. So children, that is about the seed. And where are the seeds found? They are found in the fruit. What is actually fruit? It is nothing but fertilization के बाद ovary develops into fruit. And outer egg wall, it will develop a wall which is called as the pericarp. Then in the fruits, normally we eat fruits, we know the nutrients and how they taste and all, but actually there are different types of fruits. How can you classify the fruits as fleshy fruits and dry fruits? If they are having the endocarp and mesocarp are fleshy, like guava. Guava, you see children, when you cut into inside, it is very soft. Or mango, it is very soft. They are called fleshy fruits. And the dry fruits, if you see, the groundnut or mustard, and many other, what we eat like groundnuts and others, these are called dry fruits. No fleshy or pulpy material is there inside. So, next, another classification is also there depending on the formation of a fruit. One is called a true fruit, and other is a false fruit, and one more variety, parthenocarpic fruit. So what is the difference between all them? True fruit, false fruit, and parthenocarpic fruit. True fruit, where children, ovary develops into fruit. False fruit, along with ovary, even thalamus also will be contributing in the formation of fruit. Then that is called as a false fruit. Normal fruits are all true fruits generally, whereas a false fruit, example, you can know apple, cashew, strawberry, these are all come under false fruits. And third type, I told you, parthenocarpic fruit. These fruits, children, they are formed without fertilization. Without fertilization, we are telling that after fertilization, ovary develops into fruit. So, without fertilization itself, the fruits, the ovary develops into fruit, then we call it as parthenocarpic fruit. Example, banana. Generally, many seedless fruits, generally we can see as a parthenocarpic fruits. So, these are the different types of fruits, children. One is a true fruit, false fruit, and parthenocarpic. And other classification is fleshy fruits and dry fruits. Coming to the advantages of the seed. We have learnt about the seed and all, and structure of the seed. Now, what is an advantage? Does the seed make any advantage for the plant? Definitely yes. Because first point, if you see, seeds have better adaptation strategies. You have seen already they can survive even in the dry condition by slowing down the metabolic activities, and they are protected by seed coat. So, and not only that, they have the mechanism for dispersal. Children, by wind and all, some feathery seeds will be flying. So these seeds are carried even either by animals or wind or by water. Again, they can be carried to different locations, and which are having their mechanisms for dispersal. Like how the pollen grains are carried, even the seeds are carried to other areas. So they are helping in the spreading of the species. And they have sufficient food reserves. Another advantage, what children? They are able to survive because the embryo is able to survive because it is having a sufficient food reserve. To nourish even the seedlings after germination also, you could observe a small tamarind plant or or a small neem tree which is growing, small seedling, you can see the cotyledons will be very much present. What they will be doing, they're helping even the growing plant, like parental care in animals and all. You can see the cotyledons will be caring for the developing seedling until it is capable of preparing its own food or photosynthesis. It will be helping to provide, the cotyledons will provide nutrition for the developing seedling. And seed coat, already I told you, it is protecting the embryo. And most importantly, the seeds help in variation. Because the seed is formed because of fertilization or cross-pollination and all that. Then where we can see, even in self-pollination also, but the seeds are helping in promoting variation. So that's about all advantages of seeds. 2 marks or 3 mark question. This is important, children. Advantages of seeds. Then coming to the viability of seeds. Like children, we have seen in pollen grains. What is the viability of pollen grain? How many days or how many months they could survive? The same way, viability of the seeds also few months to years. Few months to certain years, the seeds can survive depending on the type of the species. Oldest seed, this you can remember for 1 mark question, Lupinus arcticus. Lupinus arcticus, 10,000 years of dormancy, very maximum years, number of years. And another one, date palm, one variety, Phoenix dactylifera, that is 2,000 years old seeds. So this is about the viability of seeds. These 2 examples, please remember, children. And 2 important aspects here you have to learn, that is in this topic, apomixis and polyembryony. Very, very important for examination also, and these are the 2 important, very latest developments. That is apomixis. Apomixis is producing seeds without fertilization. Without fertilization, no fertilization means it is a asexual reproduction. But formation of seeds, it is a type of sexual reproduction. So, this procedure or the phenomena, it imitates sexual reproduction in producing seeds. But normally, functionally, it is a asexual reproduction. Because the seeds are developed there, but they are developed without fertilization. Very important, children. Apomixis. So what are the ways? How can we develop these apomictic seeds? In apomictic seeds development, the diploid egg. Diploid egg is diploid. That means it has to undergo meiotic division and forms mother cell, gamete mother cell should undergo and form haploid egg. But without forming the haploid egg, the diploid egg itself, without fertilization or fusion or syngamy, develops into embryo. Then it is called, it can be developed as apomictic seed. In some cases, like mango, citrus and all, you can see children nucellus, which is there in the ovule of integuments of the ovule. What happens for no reason, without no reasons not known, the nucellus will protrude into the embryo sac. They themselves, because embryo sac, around the embryo sac there is nucellus. In some fruits, some ovules, the nucellus will protrude into the embryo sac, and on their own they develop into diploid embryos. So, in that case, we can see more number of embryos. Instead of one embryo, one egg is there inside the embryo sac. But here what happens, even nucellus also entering into the embryo sac and developing as embryos. So, this condition we can say as polyembryony. So, these are the 2 ways where the diploid egg directly develops into embryo, or the new cellular cells will protrude into the embryo sac and develop into many embryos, which condition is polyembryony. So, why we are taking this apomixis into consideration? Why we are studying? What is the advantage of apomictic seeds? So, apomictic, already we have learned the seeds are formed without fertilization. It is a type of asexual reproduction that mimics sexual reproduction. So, formation advantages where we can use this apomictic seeds. Children, hybrid varieties. The farmers are going for hybrid varieties. What do you mean by hybrid varieties? You have learned in 9th class also. Hybrid varieties in plants as well as in animals. Why Green Revolution and all, if you remember, they have developed hybrid seeds and hybrid plants. What is the use? Of two different varieties, we can cross them. We are getting the desired characters of both the varieties. And we are developing a new plant with two sets of desired characters. That is called hybridization, hybrid variety. One plant is having one special character and the other variety is having another character. ये दोनों को mix करो, hybrid you will get, and you are getting both the characters of which are desired for the farmer. So these hybrid seeds, children, the farmer is nowadays going for hybrid varieties. So hybrid varieties may they have to buy the hybrid seeds. And then so you see the roses or many other small bonsai and other dwarf variety of coconuts, dwarf variety of wheat, maize, all this we are getting in the hybrid variety. So what the farmer has to do to get that variety, they have to go for hybrid seeds. So when they go for hybrid seeds, they grow the crop, they will be getting only in that season they are getting the required desired variety. If they continue for the next generation, the same seed, then they don't get the seed will be having a different characters. It will not be exactly the same variety. Suppose you take these hybrid seeds again, cross them. Again you will get segregation. You have learned in 10th class about Mendel's law of segregation. The characters will segregate. And again, there is a new variety, new variation may arise because you are going for cross-pollination. Crossing, whenever you are crossing in two different varieties, you are getting again another variation, not the exact variation which you want. So, farmers kya karna hai? For them, they have to buy the same variety of seeds again. They have to go to the distributor, go there and buy again the same variety of hybrid seeds because he cannot grow the hybrid variety on his own. Because every season they are getting the characters to be segregated. New variety, new variations are coming. So, in that is the case, the farmer has to go and buy the hybrid seeds year after year or season after season. It is becoming very expensive issue for the farmer. So, in that case, suppose you are growing the apomictic seeds. You are converting this hybrid variety into apomictic seeds. Without fertilization, if you are capable of getting the seeds year after year, season after season, the same variety you can get. Why, madam? Why you can get the same variety? What is apomixis, children? Apomixis is nothing but development of seeds without fertilization. So one variety you have got, and try to develop apomixis or polyembryony in that aspect. Then what happens? Without fertilization, you'll be getting seeds. And no fertilization means kya hoga? There is no segregation of characters. Whatever seeds, whatever you have got the plant, it retains the same characters year after year or season after season because they are getting producing the seeds without fertilization, that is by means of apomixis. So it is a great advantage to the farmer. That is why the scientists are trying to develop the hybrid varieties in the form of apomictic seeds so that the farmer need not buy the hybrid seeds every season or every year. So children, that is the importance and advantage of apomixis and polyembryony. These two aspects are very, very important for examination. You'll be getting a 3-mark question or a 2-mark question. Earlier I told you bagging and emasculation, artificial hybridization. That is also very important. Please learn, write the points and learn apomixis and polyembryony. So that's all children about. Here we completed, the small part is left. Last video I have given you only up to fertilization. So here you have learnt about post-fertilization events, in that the types of fruits you have learnt, and the embryo formation, development, embryogenesis you have learnt, and the structure of embryo you have learnt, and now the types of seeds you have learnt, and now the last topic, apomixis and polyembryony. I hope you understood, children. Once again you listen, compare with your textbook and all, then you can easily understand in a better way. Okay, please follow and the videos and learn the content. Okay, bye.